The genus Malacothamnus, also known as the bush mallows, are fire-following shrubs in the mallow family. Here you can see some variation in the size and shape of Malacothamnus plants. Most species are around 3 to 7 feet tall, but larger plants of a few species may be up to 18 feet tall. The primary distribution of Malacothamnus is in the California Floristic Province, with some outliers in Arizona and possibly Sonora, Mexico. They occur from near sea level to roughly 9,000 feet in elevation, with the highest elevation being on the eastern side of the Sierra Nevada range. Malacothamnus generally germinate only after fires, though sporadic plants may germinate with other disturbances such as roadside grading. Most plants only live 3 to 20 years, with the majority dying in the first 10 years after a fire, often due to competition with other returning shrubs. Mean fire return intervals where Malacothamnus occurs are 10 to 150 years, which suggests seeds are likely viable for 100 plus years in at least some of those areas. Germination tests at California Botanic Garden Seed Bank confirmed at least some seed from herbarium specimens are viable after 88 years. In the bottom photo, you can see a burn boundary, which well illustrates the fire-following life history of Malacothamnus. The area shown on the left side of the photo likely has not burned for decades. That area is dominated by pines, and there are no Malacothamnus to be found. The area on the right side of the photo burned 2 years before this photo was taken. All the small green shrubs you see there are Malacothamnus. The Malacothamnus seed that was lying dormant for many years in the soil germinated after the fire and became the dominant species in this area. Many of these Malacothamnus plants will start dying out after just a few years, and all or most Malacothamnus plants will ultimately die back and make way for other vegetation and then wait in the form of seed for the next fire, which might not come for another 100 years. Let's take a look at some of the morphological variation in the genus. Trichomes are a very important character in distinguishing taxa in Malacothamnus. Here you can see a comparison of trichomes on the stems of 4 species, which shows some of the variation in trichome size and density seen in the genus. These are scanning electron microscopy images comparing 2 size extremes of stellate trichomes in Malacothamnus. The branches at the tip of the stellate trichomes are called rays, which I measured to the nearest 1/10 of a millimeter for my morphological analyses. You can see on the right that some of the stellate trichomes can have stalks. In these plants where the stellate trichomes do have stalks, they're generally mixed in with other trichomes that do not. Here's another scanning electron microscopy image showing both stellate trichomes and glandular trichomes. Stellate trichomes are the white ones with many branches. The glandular trichomes are the ones that are colored somewhat reddish. In most Malacothamnus taxa, the glandular trichomes are much smaller than the stellate trichomes. In a few taxa, like this one, the glandular trichomes are much larger, making this a very useful character in distinguishing these taxa. Glandular trichomes have been overlooked in past treatments, likely because they are so small. Note that the especially larger glandular trichomes shown here are still only about 3/10 of a millimeter long, so they are easy to overlook. The calyx and bracts are also very important characters in distinguishing Malacothamnus taxa. The calyces of Malacothamnus are fused at the base into a tube. Above the tube are lobes of various shapes. Note the variation in the shapes of these lobes. Some are more triangular, with the lobe widest at the base. Others are more rounded, with the lobe widest near the middle. The arrows here point to calyx bracts, which are a whorl of 3 bracts beneath each calyx. The size and shape of these and how long they are relative to the calyx is also important in identification. Here you can see more examples of variation in the calyx bract. Another type of bract useful in identification are stipular bracts. The stipular bracts are found in the inflorescence between the calyx bracts and the leaves. The size and shape of the stipular bracts is very useful in distinguishing a few taxa. Stipular bracts are another character not previously assessed by other researchers. In most Malacothamnus taxa, the corollas dry closed and somewhat twisted, as you can see on the right. In a few taxa, though, they dry open, as you can see on the left. Why they do that, I have no idea. That is perhaps something a future researcher can look into, though. This is a character that I discovered while doing field surveys of the genus, which shows the importance of not relying entirely on herbarium specimens. Inflorescences in Malacothamnus can vary from head-like to spike-like to panicle-like. There are some intermediate forms as well, and some taxa can have multiple inflorescence types. Leaf shape in Malacothamnus could be highly variable, even on the same plant. Two leaf characters that are somewhat useful in distinguishing taxa are the degree of lobing and the shape of the leaf base. You can see here that leaves vary from unlobed to slightly to moderately lobed, and that the leaf bases vary from cuneate to truncate to cordate. Now let's take a brief look at the taxonomic history of the genus. Malacothamnus was first collected in 1836 and included within the genus Malva. In 1849, the genus Malvaceae, which included what is now Malacothamnus, was split off of Malva. In 1891, Malvaceae was briefly changed to Malvaceae as the name Malvaceae had taxonomic priority over Malvaceae, but the name Malvaceae was conserved shortly thereafter. And in 1906, Malacothamnus was finally described. E.L. Green was the person to describe Malacothamnus, and he distinguished it from Malvaceae based on its shrubby habit and some differences in its fruit. On the upper right, you can see a full Malacothamnus fruit with its many carpels. Below is a single carpel. Note that the carpels in Malacothamnus completely split into two halves as shown on the bottom right. In some other Malogenera, they only partially dehisce. In Malacothamnus, there is no reticulation on the carpel surface, which you can see in the photo here. In some other Malogenera, you may see a net-like patterning or bumps on the carpel surface. Malacothamnus also only have a single seed per carpel. Even though the name Malacothamnus was published over 100 years ago, not everyone agreed that it should be recognized as a separate genus. For many years, most authors still included it in Malvaceae, and a couple authors even lumped Malvaceae into Spiralcea. In 1951, Thomas Kearney published the first treatment to consider all taxa currently in Malacothamnus, and with a couple minor exceptions, this was the standard Malacothamnus treatment for the next 4 decades. In the 1990s, things got messy though. David Bates published his Malacothamnus treatment for the first edition of the Jepson Manual. In this treatment, he lumped over half of Kearney's taxa, including 10 rare taxa. This caused a lot of controversy as far as how many taxa should be recognized, as Bates himself admitted that Kearney's treatment was equally justifiable to his own. This led to further research by Tracy Slaughter, who tried to provide more evidence for which taxa should or should not be recognized. She was the first person to do both morphological and phylogenetic analyses on the full genus, as well as the related genus Iliamna. Her phylogenetic analyses confirmed that Malacothamnus is monophyletic, and that it is closely related to the genus Phimosea and part of Iliamna, which had been previously proposed by Bates as closely related to Malacothamnus based on chromosome counts. ITS, however, did not resolve any relationships within Malacothamnus. Slaughter did, however, find morphological evidence that disagreed with some of Bates' lumpings, so she split back out 6 taxa in her treatment. When I first started looking into Malacothamnus in 2015, I found 3 conflicting treatments. Bates' Flora of North America treatment had just been released, which is almost identical to his Jepson treatment and only recognizes 11 taxa. Slaughter's treatment had been recently published in the 2nd edition of the Jepson Manual, recognizing 17 taxa. And the California Native Plant Society Rare Plant Inventory mostly followed the 1951 Kearney treatment, recognizing roughly 26 taxa. 6 to 10 of the rare taxa recognized by CNPS were not recognized in the other treatments. One problem with all these treatments is that no author shows adequate evidence to support their treatment. I wanted to remedy that. So let's move on to my research. My primary research goal was to clarify the taxon boundaries within Malacothamnus for conservation purposes. I wanted to provide evidence showing which taxa should be recognized, how to distinguish them from each other, and to document where they do or do not occur. I used 3 main lines of evidence in my research: morphological analyses, phylogenetic analyses, and reproductive barriers. For my morphological analyses, I refined and added to the characters measured by Slaughter. I also doubled the sampling of specimens she measured. For my phylogenetic analyses, I used RADseq, which results in thousands of more loci than the Sanger sequencing used by Slaughter. For these analyses, I sample about 10 times as many specimens as her. Regarding reproductive barriers, one major contribution by Bates is that he did some crossing studies back in the 1960s and found that all Malacothamnus can likely hybridize, and that they produce fertile hybrids. So possibly the only reproductive barriers in the genus are geographic isolation and flowering time. There are lots of possible hypotheses regarding which taxa within Malacothamnus should be recognized. At the most basic level, the circumscription of every basionym, which is the first name used for each described taxon, could be considered a hypothesis of a lineage distinct enough to be recognized. Each basionym currently included in Malacothamnus is shown here as a solid black circle. On the far left, you can see there are 36 of these. These are 36 described taxa, excluding all synonyms. So these represent the finest level taxon hypotheses that have been proposed. The center part of the slide represents how Kearney treated each of these basionyms. Note that some of these basionyms are contained within blue circles. Each blue circle represents where Kearney lumped 2 or 3 basionyms into a single taxon. Each of these lumpings represent the hypothesis that the lumped taxa are more closely related to each other than to the rest of the genus. On the right, the yellow circles represent further lumping done by Bates. Bates lumpings represent the broadest hypotheses of what should be considered a single taxon in Malacothamnus. These again represent the hypotheses that the taxa lumped together are more closely related to each other than to the rest of the genus. Bates lumpings are morphologically based, so he is also offering the hypothesis that taxa that are more morphologically similar to each other are more closely related. Shown in pink are the 5 varieties of Malacothamnus fasciculatus recognized by Kearney. This shows the hypothesis that these 5 lineages are more closely related to each other than to the rest of the genus. Bates' idea of Malacothamnus fasciculatus lumped these 5 varieties with 4 other Kearney taxa, which provides the hypothesis that this larger grouping of taxa are more closely related to each other than the rest of the genus. So you can see there are levels upon levels of hypotheses to investigate. Another hypothesis to test is that disjunct populations of a single taxon could be morphologically and/or phylogenetically distinct. Shown here are 2 suspiciously disjunct regions of Malacothamnus davidsonii. These could be closely related, as maybe a bird could have dropped some seed from one area to another and established a new population that way. Alternatively, there could be convergent evolution going on in which 2 unrelated taxa converge morphologically. Specimens problematic to identify could perhaps be distinct as well. Shown on the right is part of a 1976 Santa Barbara regional flora that indicated a possible undescribed species that hasn't been previously assessed before my study. Additionally, it's been proposed that some taxa integrate morphologically. These too are hypotheses that no evidence has been provided for. Whether these taxa actually integrate and to what degree needs to be confirmed. So now that I had my hypotheses, I needed to collect data and analyze it. Over the past 7 years, I've done roughly 350 focus surveys for Malacothamnus, covering most of its known range. On these, I collected both voucher specimens and tissue samples for morphological and phylogenetic analyses. I mapped the distribution of Malacothamnus wherever I found it, with special attention to trying to find the edges of ranges, hybrid zones, and to fill in gaps in the known ranges. I also collected additional information that is often not included on herbarium specimens, such as plant height and the flowering phenology of the population as a whole. For the morphological analyses, I measured 41 characters on 773 specimens and tried to measure roughly 20 specimens per hypothesized grouping when enough specimens were available. About half of the characters I measured were related to trichomes. I used 3 types of morphological analyses. I used principal component analysis, or PCA, for data exploration, and to test how morphologically distinct the various hypothesized groupings were. You can see in the above PCA, the 3 colors represent the hypothesis of 3 morphological groups. The resulting character loadings from the PCAs also clarify which measured characters are causing the separation seen in the plots, which can be useful in writing keys to distinguish them. I used linear discriminant analysis to test how well the characters I measured can predict the assigned morphological groupings. In the example PCA above, the analysis would likely predict most of the proposed groupings correctly, but possibly have problems where the red and cyan groups overlap. I also used pairwise permutational multivariate analysis of variance, or PERMANOVA, to test the statistical significance of differences between each assigned group based on the morphological characters measured. This is essentially where I get my p-values from that support the groups that I found in my PCAs. For the phylogenetic analyses, I used a CTAB-based protocol to extract nuclear DNA from roughly 360 Malacothamnus samples and 11 outgroup taxa. The tubes shown in the image are some samples partway through the DNA extraction process. For my RADseq library preparation, I used a single digest with a PST1 restriction enzyme. And after sequencing, the sequences were assembled using IPyRAD with mostly default settings. I primarily use 4 types of phylogenetic analyses. I use maximum likelihood and quartets-based species tree analyses to assess relationships between samples within a bifurcating tree. I also use neighbor net analyses and multidimensional scaling to further explore some relationships outside the confines of bifurcating trees, which helps assess what may be causing conflicting patterns and poor bootstrap support in the bifurcating trees. In preparation for writing a new treatment for the genus, I also reviewed over 1,000 herbarium specimens, over 9,000 iNaturalist and CalFlora observations, and rare plant records in the California Natural Diversity Database. Once I had all my evidence, I needed to make decisions on how to delineate taxa. To do that, it is good to follow a species concept. I'm using the unified species concept of Keir's and its companion subspecies concept. The species concept says species are independently evolving metapopulation lineages, and that subspecies and varieties are incompletely separated lineages within a more inclusive lineage. As you can see in the illustration on the right, when you have enough evidence to prove that you have independently evolving lineages, you can call them multiple species. If you have evidence that indicates diverging lineages that are very closely related, or there are perhaps some ambiguities in your evidence, it may be better to consider those taxa as subspecies or varieties. Note that subspecies and varieties are often used interchangeably in plant taxonomy, so there's arguably no difference between them unless they are both used in the same name. I'm not going to complicate matters by doing that, and I just use varieties to be consistent with most previously named taxa in Malacothamnus. Let's take a look at some of the broad scale results before delving into some more specific examples of what I found. My morphological PCAs resulted in 38 groups with both morphological and geographic cohesion. These vary greatly in distinctness, with some clearly being both morphologically and geographically distinct, and others being more ambiguous. I wanted to be very conservative in the groupings I chose in my PCAs, as these are hypotheses I would test with phylogenetic analyses. So while some of these groupings are very distinct, some may just be diverging populations within a single taxon or hybrid populations. You can see on the right a morphological distance matrix that summarizes how similar each of these groups are to each other. The darker blue squares represent pairs of morphological groups that are more similar to each other, and the darker reds represent morphological groups that are less similar to each other. The bold black square surrounds all groups lumped into Malacothamnus fasciculatus by Bates, which was the largest lumping of taxa done in the genus. You can see a lot of blue here, which supports at least some degree of morphological similarity between these groups, though some are much more similar than others. Permanova results show that differences between all these groups are statistically significant, with the exception of those that were inadequately sampled. A few groups only had 2 or 3 samples, which really aren't enough to assess statistically. Using linear discriminant analysis to test how well the characters I measured could predict the groups I assigned the specimens to had mixed results. It was able to predict many morphological groups fairly well, but wasn't particularly accurate in distinguishing some of the more ambiguous morphological groupings, which is to be expected. I assembled a variety of alignments using IPyRAD, with 3 focusing on the full genus. One contained everything, including likely hybrids. A second was similar, but I removed all likely hybrids that were not part of one of the 38 groups from the morphological analyses. These larger alignments had a mean of 7,000 to 8,000 loci per sample and about 90% missing data. Missing data is one of the downsides of using RADseq. RADseq gives you a huge amount of data, but a lot of missing data as well. The image below shows you a glimpse of a tiny part of one alignment of RADseq data to help you visualize what the missing data looks like. Because RADseq provides a lot of data, and some types of analyses can work around the missing data, 90% missing data is not as bad as it sounds. That said, it's nice to have as little missing data as possible. To test the effects of missing data, I also used a reduced set of samples with especially high numbers of reads to produce an alignment with only 77% missing data and 2 to 3 times as many loci as those from the larger alignment. Bootstrap support was mixed. You can see here one of the larger maximum likelihood trees on the left and the reduced data set on the right. What you see in purples and darker blues are where there's pretty good bootstrap support in these trees. Where you see greens and yellows, there's poor support. Those parts of the tree with poor support are mostly in areas where there's likely more gene flow. This may be within a single taxon or between geographically adjacent taxa. Low bootstrap support could also indicate incomplete lineage sorting. Note that the reduced dataset on the right does have overall higher bootstrap support than the larger dataset, but still has some problematic clades, and these clades correspond to where there is known hybridization between taxa. Here I show a tangogram comparing the maximum likelihood tree of the reduced dataset to the quartet-based species tree. The gray-colored clades are those generally corresponding to morphological groups, and you can see that they mostly correspond between trees. Relationships between clades mostly correspond, but there are some differences between each tree. There are clear geographic patterns throughout the phylogenetic analyses. At the broadest level, there is always a northern clade shown in blue, and a southern clade shown in orange and red. The southern clade is always further divided into northern and southern subclades, as seen by the orange and the red. In yellow, you can see two clades that are mostly geographically intermediate between the northern and southern clades. In some analyses, these are a single clade. In others, like this one, they form two clades, each sister to either the northern or southern clade. Here we are looking at 6 species as classified by Bates. Bates was the person that did the most lumping of taxa in the genus. It's important to note that Bates' species can be viewed as hypotheses that the morphologically similar taxa he lumped together are closely related, which clearly isn't the case. While there are some morphological patterns seen in the phylogenies, the relationships between taxa shown in the phylogenies are generally more geographically cohesive than morphologically cohesive. These trees really show how major morphological characters used in identification in the genus come out all over the phylogeny. It also shows that some of Bates' taxa are exceedingly overlumped. Some of the polyphyly in the phylogenies isn't well supported, though. Gene flow or incomplete lineage sorting in some regions proved problematic for some analyses. The areas circled in red are one of the areas in the phylogeny with very low bootstrap support and show possible gene flow between Malacothamnus densiflorus and 2 other species around San Diego County. Each of these species is mostly morphologically and geographically distinct, but there are small hybrid zones where the ranges come together. 5 of Bates' species turned out to be solidly monophyletic, so the 3 shown at the bottom are probably only so because he didn't lump them in with any other species. Bates did find a couple morphological patterns that corresponded to clades, though. Malacothamnus palmeri in the upper left was recognized as 3 varieties by Keir and are distinguished from the rest of the genus by generally having a capitate inflorescence. This turns out to be one of the few morphological characters that on its own can mostly be attributed to a single clade. Malacothamnus jonesii on the upper right was recognized as 3 species by Keir. These taxa have very similar leaf shape and trichomes, which makes them often indistinguishable when in flower. So the combination of several morphological characters does accurately reflect phylogenetic relationships in this clade. If we look at the morphological groups from my PCAs on a phylogeny, you can see here that 16 of them are monophyletic. These are shown as the spiky purple clades. 9 more morphological groups fall out into the 3 spiky orange clades. These groupings mostly sort out within the clades, but are not monophyletic. These blue clades represent a single morphological group, which was recognized as a variety of Malacothamnus fasciculatus by Keir. We'll take a brief look at that later. What remains in white are 9 somewhat messy groups with possible gene flow and/or incomplete lineage sorting. The larger red clade includes plants from San Diego County, California, and Baja California, Mexico, and is the major area of the tree with low bootstrap support. Hybrids are common where each of these taxa meet, so it isn't too surprising that the phylogeny is a bit messy here. Now let's take a look at some of the finer scale results. As mentioned before, the Malacothamnus palmeri varieties can be separated from the rest of the genus by generally capitate inflorescences, as you can see here. The main question I had regarding them was whether they integrated or not, and to what degree. All previous researchers said that they integrate. Turns out that may not be true. The morphological PCA you see on the upper right shows 3 distinct groups, each with its own geographic range, as seen on the left. These align with 3 closely related clades in the phylogeny. It is possible that there may be intermediates where the ranges come together, assuming they actually do, but there's no evidence of this at this point. As these 3 groups are distinct both morphologically and geographically, and form distinct clades, I'm treating them as species. You can see here some of the distinguishing characters between them. Stipular bracts are shown above, and density of trichomes on the leaves is shown below. If you are especially interested in this group of 3 species, I published a focused morphological study on them in 2021. which is linked to on my website. Bates' idea of Malacothamnus aboriginum consisted of two very disjunct regions of plants that are united by the character of relatively wide stipular bracts. These are clearly distinct, though. The PCA shows them to be morphologically distinct, and the phylogeny shows them not to be closely related at all. Note that the scion plants are polyphyletic. This is presumably due to gene flow or incomplete lineage sorting with the adjacent Malacothamnus densiflorus, with which it does hybridize with, where the ranges meet. Tom Chester and I published these southern populations as the new species Malacothamnus enigmaticus in 2019. As shown in the photos, these two species can easily be differentiated by the length of the stellate trichomes on the stem, and there are many other characters that distinguish them as well. The paper describing Malacothamnus enigmaticus is also linked to on our website. Malacothamnus davidsonii, as recognized by all previous authors, also consists of plants from two disjunct regions. Not only are plants from these two regions morphologically very similar, but they also bloom relatively late compared to most other species, which provides further evidence that they could be a single species. If you look at the PCA here, you can see that there is a clear geographically based pattern to the morphology, but they are far from distinct. Also note that the blue triangle represents the type specimen, which could arguably be placed in either group. If we look at the phylogeny, however, plants from these two regions appear to not be closely related at all. So this could represent convergent evolution of two unrelated taxa. Alternatively, perhaps these two taxa shared a relatively recent common ancestor, but gene flow with geographically adjacent taxa has mass signal of this in the phylogenetic analyses. I could find no evidence of that in my analyses, though. Either way, the red population represents a morphologically cryptic new species, which will be published soon. Another example of a species with disjunct populations is Malacothamnus orbiculatus, which was discovered in Arizona and a few places in recent years. This PCA represents a good example of not finding any pattern at all. Plants from Arizona and California are both highly morphologically variable, and this variation seems to be mostly consistent between them. Phylogenetic analyses place the Arizona plants within a single clade that is always within the California clade, but moves around within this clade in different analyses. This disjunction implies that Malacothamnus orbiculatus was possibly once more widespread in what is now the Mojave Desert, and that populations may still be found in desert mountains where they are currently unknown. Malacothamnus densiflorus has two varieties that have always been a source of confusion. After looking at specimens, I hypothesized that trichomes could tell them apart. Here you can see variety densiflorus on the left has fairly sparse stellate trichomes on the calyx tube, and variety visitus on the right has pretty dense stellate trichomes there. Another major difference is that what I hypothesized to be variety visitus has much longer glandular trichomes than variety densiflorus. In the past, these varieties have been distinguished from each other based on the length of the calyx and calyx bracts. Trichomes were not considered as useful in distinguishing them. So I wanted to compare PCAs with and without trichomes. Specimens in both of these PCAs were classified using the key of Kearney from the 1950s. On the left, you can see a PCA without trichomes, which arguably supports the hypothesis of two integrating varieties. On the right is a PCA with the trichomes included, and you can see two groups are formed that are very distinct and do not follow the previous classification at all. This really shows the importance of reassessing past morphological hypotheses. By adding additional data, I got completely different results. The characters used in the past to differentiate these varieties essentially just differentiated more and less robust plants from each other. The picture from the phylogenetic analyses, however, is not so clear. Most of Malacothamnus densiflorus is in the messy San Diego County clade with low bootstrap support. The clade in blue I've circled is the type locality of Malacothamnus densiflorus, which possibly complicates things further. These are within a clade of Malacothamnus fasciculatus variety laxiflorus, which they grow adjacent to. So gene flow between adjacent populations of different taxa appears to be totally confounding the phylogenetic analyses. While the phylogenetic analyses are messy, the Malacothamnus densiflorus varieties are two of the most morphologically distinct taxa in the genus and show clear geographical patterns that correspond with the morphological patterns. When I ran a PCA of the entire genus, the Malacothamnus densiflorus varieties were the second and third groups to fall out as distinct from the rest. Based on this, I'm following the morphological and geographic evidence and recognizing these as two distinct taxa. Arguably, they could be recognized as species, but as the phylogenetic analyses are messy, I'm keeping them as varieties until there is more solid evidence about their relationships. Bates' idea of Malacothamnus fremontii contains 4 species as defined by Kearney, and 2 possible undescribed species noted by other researchers. As you can see, this grouping of taxa is polyphyletic. The cyan new species and Malacothamnus orbiculatus in yellow are in different clades from the rest, and the red new species is in a small clade that is sister to the purple clade. Looking at the morphological PCA, you can see the new species colored cyan is clearly distinct. The new species colored red is mostly distinct here and completely distinct in pairwise PCAs focused on the groups shown overlapping here. One group within the purple Malacothamnus fremontii clade is morphologically distinct. And Malacothamnus orbiculatus in yellow overlaps with the rest of the Malacothamnus fremontii clade in morphological analyses. While Malacothamnus fremontii and Malacothamnus orbiculatus do look very similar sometimes, they can easily be distinguished by the way the flowers dry, as seen in the photos here. All taxa in the purple Malacothamnus fremontii clade have the flowers drying open. Malacothamnus orbiculatus and the cyan new species have flowers drying closed. The red new species, which is closely related to the Malacothamnus fremontii clade, has flowers drying somewhat open, which suggests maybe past gene flow between them or a common ancestor with flowers that dried open. The character of flowers drying open is the second of only 2 morphological characters that by themselves are mostly confined to a single clade, the other being the capitate inflorescences of the Malacothamnus palmeri clade. Looking at the map again, you can see that there is geographic cohesion in the purple Malacothamnus fremontii clade, and that possible gene flow with the new species shown in red makes sense geographically as well. Now let's take a closer look at what is going on inside the purple clade. This is still the purple clade, but now divided into other colors. In my morphological analyses, I defined 4 morphological and geographic groupings. The cyan group is the one that came out distinct in the previous PCA I showed. In this PCA, you can see that the red and yellow groups are mostly distinct and maybe intergrade, which makes sense based on their geographic pattern. The orange group, however, is morphologically intermediate between the red and yellow groups, but somewhat disjunct from both geographically. This made me curious about it, so I retained it for the phylogenetic analyses. On the bottom left is a neighbor net analysis of the same groups. You can see that the cyan, red, and yellow groups each form a nice clade, but the orange group again appears to be maybe intermediate between them. So it's not really clear what is going on here. I think more work needs to be done in this clade with much better sampling, as there are a lot of gaps in the distribution sample. In the meantime, I'm recognizing everything you see here as Malacothamnus fremontii and distinguishing the cyan group from the rest as a variety. Someone else will have to take a closer look at this clade to see if any of the other groupings should be recognized with a taxonomic rank. I almost recognized the red and yellow groups as different taxa, but couldn't devise a morphological key that could accurately distinguish them, and later found many specimens that weren't included in the analyses were possibly intermediate between them. Malacothamnus fasciculatus, is recognized by Bates, is the biggest lumping of taxa in the genus, and is mostly differentiated from the rest by the combination of relatively short calyx bracts and short stellate trichomes, as shown here. Bates' hypothesis that these taxa should all be considered one taxon does not hold up, though, as they are scattered throughout the larger northern, central, and southern clades. Let's take a closer look at the northern groupings that are disjunct from the rest. You can see on the right that there are some relatively clear morphological patterns distinguishing the northern plants, and that these match up with previously described species. Looking back at the phylogeny, these northern species also hold up as clades. More complicated is everything shown here in pink, which is what Kearney recognized as Malacothamnus fasciculatus. Let's zoom in on that. Shown in pink is still Malacothamnus fasciculatus. The other colors on the map represent other species that are sister to the various Malacothamnus fasciculatus clades. I've shown these on the phylogeny as well, and colored each clade to show which species the various clades of Malacothamnus fasciculatus are sister to. The specimens in each clade are outlined in the same color on the map. Note that each of the other species is unquestionably distinct from Malacothamnus fasciculatus based on morphology. So arguably, if I were to go with the phylogenetic evidence here, each of those Malacothamnus fasciculatus clades could be recognized as a distinct species, even though they are very similar morphologically. I wondered, though, if perhaps gene flow with Malacothamnus fasciculatus and geographically adjacent species could perhaps be confounding the phylogenetic analyses. One test I thought of was to run a maximum likelihood tree of just the Malacothamnus fasciculatus varieties to see if I got the same relationships between the Malacothamnus fasciculatus clades. This is shown on the left. I've colored the clades on the inside of the circle the same for both phylogenies. Note that the single cyan clade from the full genus phylogeny on the right is broken up into 3 clades on the left phylogeny of just Malacothamnus fasciculatus. So these two trees do not agree with each other regarding those clades shown as cyan. I did many other analyses, which I don't have time to go into, but in addition to possible gene flow between adjacent species, there appears to be geographic patterns of gene flow between the Malacothamnus fasciculatus clades as well. Ultimately, more work needs to be done here, probably with population-level sampling. As the phylogenetic evidence isn't especially clear for these pink clades, I'm mostly defaulting to the past varieties of Malacothamnus fasciculatus recognized by Kearney, which make both morphological and geographic sense. The one exception is the Malacothamnus fasciculatus in the green clade. This is Malacothamnus fasciculatus variety Nutalii, which is the most phylogenetically and morphologically distinct of the varieties. It also has a peak bloom time about 1 month later than the rest. Based on these lines of evidence, I think it's safe to recognize that clade at the species rank. So those were just some examples of the analyses I ran. Now let's take a look at the primary conclusions from all of the analyses. Here we see the comparison of minimum rank taxa in the Kearney and Bates treatments from earlier, with the addition of the taxa I will recognize on the right. You can see the taxa I will recognize are mostly the same as Kearney, with a few exceptions. At the bottom right are 4 pink circles. These are the 4 new species I will recognize. Additionally, I accept one of the lumpings done by Bates in yellow and lump 2 other taxa of Kearney's. Both of these lumpings that are different from Kearney need further research and could be split back out with further evidence. Let's look at my final taxa from a phylogenetic perspective. I'm recognizing 14 monophyletic species, each without varieties, shown by the spiky purple clades. The orange spiky clades will be 3 monophyletic species, each with 2 to 3 varieties. Some or all of those varieties could arguably be recognized as species, but some ambiguous regions within these species make varieties perhaps more appropriate. The remaining white areas of the tree contain 5 polyphyletic species, with 0 to 4 varieties each. It is worth noting that while not monophyletic, many of these clades are mostly one taxon, and that gene flow and/or incomplete lineage sorting is probably what is making things messy. There are 2 monophyletic varieties here, and these are the 2 island varieties of Malacothamnus fasciculatus, which are more geographically isolated from other taxa than the rest. Thus, relatively recent gene flow is less likely with them. Despite the phylogenetic evidence being somewhat messy in the white clades, there is good morphological and geographic evidence to fall back on, but more research should be done, especially in Baja California, where my sampling was limited. Comparing the numbers between Kearney and I, we both recognize roughly the same number of species and varieties, but there are some major differences. I add 4 new species. I change 4 of Kearney's varieties to species. I change 7 of his species to varieties, and I lump 2 of his species into other taxa. So I'm kind of walking the line between being a lumper and splitter here. Really, I'm just following the evidence. I have some general observations about this study. First, using both previously unassessed morphological characters and greater sampling of specimens was very useful in preparing morphological hypotheses to assess phylogenetically. This showed where there really was intergradation or not. It confirmed morphologically distinct new species, and it showed that some of the previous taxon delimitations were very wrong. I wouldn't have felt great about going into phylogenetic analyses without doing this first. Second, using RADseq data for the DNA analyses did way better than ITS, but was still problematic for samples from geographic regions with likely gene flow. Population-level sampling using RADseq could possibly resolve some of the questions not answered by my analyses. Likewise, using a technique like HybeSeq that has much less missing data could allow for the use of different analyses that may also help answer some of the remaining questions. Third, I found using multiple lines of evidence was very useful. In some cases, all of the evidence agreed, which was great. In other cases, some of the evidence was ambiguous. It was nice to be able to fall back on fairly solid morphological and geographic evidence when the phylogenetic evidence was not clear. Likewise, phylogenetic evidence confirmed a new species that is morphologically cryptic, which I probably wouldn't have felt justified in recognizing without that evidence. And finally, it's clear that more research needs to be done on the genus. My research answered a lot of questions, but not all of them. It did, however, clarify where more work is needed. And I'll be curious to see what the results are when future researchers tackle the remaining questions. The big product of all these analyses will be a new treatment of the full genus. This will include an identification key with character explanations and accompanying photos. I will include photos of all the taxa. On the right, you can see an example set of photos from the treatment for one of the taxa. It will also include range maps, life history, conservation concerns, preliminary conservation assessments for each taxon, as well as other details such as common names, phenology, and known hybrids with each taxon. With that, I'd like to thank everyone who has helped with my research. If you count all of the iNaturalist observations I've used, thousands of people have contributed. So thanks to everyone who has helped one way or another. If you'd like more information on Malacothamnus, please visit my website. As of now, you can find some draft regional keys to the genus, as well as links to papers that are already published. I'll add more as I finish up each piece of this research. You can expect a new treatment of the full genus and a new Jepson manual treatment sometime in 2023. Thanks very much.